Hi everyone, my name is Bryan and I'm a graduate student at the University of Washington. Today I will present about the population genetic structure of eelgrass across Washington state. Zostera marina, commonly known as eelgrass, is a bed-forming seagrass widely distributed across the Salish Sea. These beds form the foundation of estuarine habitats and provide several benefits to coastal areas and human livelihoods, including shoreline protection, food web support, and nursery habitat. Because of this recognized importance, a considerable amount of effort is dedicated to eelgrass conservation, including large-scale translocations of plants to mitigate local declines. However, all these efforts are hampered by a lack of information about the population genetic structure of local eelgrass. In the absence of such data, all plants are treated as equal, without addressing whether the genetic diversity is unique, which sites best serve as donors for transplants, or how would such transplants affect current population diversity and structure. A key first step towards improving the outcomes of conservation strategies is to determine baseline population genetic structure information relevant to the designation of management units. So, to fill in this critical knowledge gap, my research aims to describe the population genetic structure of native eelgrass populations across Washington state. By conducting this work, I will be able to generate a detailed map of genetic variation across the state that will in turn enable us to inform management by identifying populations with unique genetic diversity to protect or suitable donor sites to propagate. For this work, we sampled 16 populations. 14 of them are located in the Salish Sea and 2 on the coast. These sites are located in 4 different areas: the Pacific Coast, north of Puget Sound, the Hood Canal, and south Puget Sound. We use next-generation sequencing approaches, or RAT-seq specifically, and identified 214 variable genetic markers, or SNPs, which are single nucleotide variations along the DNA. Our initial data set contained 753 individuals, but only 436 remained after filtering out clones. This is a common practice in the study of asexually reproducing species like eelgrass to avoid confounded results in the study of diversity at the population level. This data allowed us to analyze shared genetic variation by conducting a discriminant analysis of principal components that clusters individuals based on genetic similarity. Here, each dot represents a sample, and it's colored by population. The closer 2 points are to each other in the graph space, the more genetic variation they share, or in other words, they are more genetically similar. In this graph, we can identify 4 main clusters of diversity. The most evident one is formed by the populations from the coast, circled in blue, north of Puget Sound, circled in red, and south Puget Sound, circled in orange, are genetically divergent. And similarly, geographically isolated populations in the Hood Canal, circled in purple, are also genetically isolated from the rest of the surrounding areas. Here, the x-axis that explains the most variation in the data separates the coastal and Puget Sound clusters to opposite ends. The order of the populations on the y-axis instead only partially conforms to a north to south direction for the Puget Sound clusters. We observed extensive genetic structuring at the regional level that reflects geographic spatial patterns. Our data suggests high connectivity within areas but not between them, especially between North and South Puget Sound, but also in relation to populations in the Hood Canal. This type of structuring within the same body of water can be caused by barriers to gene flow, such as currents, or distances simply too long for seed dispersal to occur successfully. We also observed that eelgrass in the Salish Sea is genetically divergent from the Washington coast. However, our analysis only contains sites from the United States, and the diversity of eelgrass across the border still remains to be described. We also observed varying rates of clonality among our populations, from 0 to 90% of samples being clones within sites. 4 of them were dominated by clones. However, we're still analyzing this data to understand exactly how the surprisingly high rates of clonality influence population and structure and therefore contribute to overall ecosystem stability. Thank you for your attention.